This conference will now be recorded.
Good evening, everyone. I am Rajni Jain. On behalf of agricultureinformation.com, I like to welcome you all to this online meeting. Please take a moment to review the guidelines for this online meeting posted in the chat window. I now request the moderator, Mr. Lakshmipati, to introduce the speaker and start the meeting. Thank you.
Thank you, Mr. Rajani. Good evening, everyone. I am Lakshmipati and I'll be moderating this meeting. Agricultureinformation.com likes to welcome to this meeting Dr. Sivalingam Elayabalan, the Technical Director and Agriculture Scientist at Sankar Biotech, Hosur, Tamilnadu. Dr. Sivalingam Elayabalan will be speaking on the topic plant propagation technology for banana and other crops. I now request the speaker to begin.
Yeah. So very good evening to everyone. Yeah. So we are the company Sankar Biotechnology. So basically, so he's the company director Sankar and myself technical director of the company. So we are working past 15 years in the field of plant tissue culture technology, especially for propagation of banana and other ornamental crops. Okay, our company is located in the central core of the Hosur, within 1 year, 1 hour journey from Bangalore to our company. So we are basically agri graduates. Okay, I'm a doctorate. So we have the hands-on experience. Apart from the hands-on experience, we have the scientific knowledge behind plant tissue culture. So both this hands-on experience and the science we are applying to propagating a plants in different kind of plants. Okay, so in this section, so I'm going to detail what we are doing now. Okay, what is the expectation of the farmers, why we want to do tissue culture technology. So our company main product is the plant-based product. Okay, so the main agriculture input is seeds and seedlings. Okay, so this is our company location. So before that, in agriculture, the foremost important is the agriculture input is seeds. That seed should be a disease-free and quality of seeds. Now the farmers are facing a big problem for getting the quality of seeds as well as quality of planting material. It's a big challenging for the production of and productivity of our agriculture crops as well as horticulture crops. The production and productivity of the horticulture crops and agriculture crops is determining based on the what they having the seed material and seedling materials. Based on that only we are predicting the yield parameters. Okay, we know most of the agriculture crops they are doing through seed. Seed is the starting material most of the agriculture crops. Okay, in horticulture crops, as we know in horticulture crops, the propagation materials are tubers, bulb, corm, rhizome. Also, most of the horticulture crops they are not having seeds. Only through they are going for cuttings. For example, in stolen, for the strawberry fruit, what we are eating, the strawberry fruit, the fruit is not having seeds. The strawberry is propagating through stolons. Okay. Also, the floriculture industry, most of the floriculture industry, they are using bulb. Also, in orchids, one of the highest commercial important floriculture crop is orchids. The orchids also propagating through flower stalks. From the flower stalks, they're multiplying. Okay, this another part. In conventionally, the propagation material are associated with fungal and bacterial, and most important is viral disease. The viral disease are for most important. It is also associated with the planting material. So the pathogens, the plant pathogens are interfering the growth and development of the plants as well as yield. Okay, why we want to go for the plant tissue culture technology? Why we want to grow your plants in the container? Why we want to grow your plants in the separate incubation chamber like intensive care unit? How we are once undergoing operations, we are keeping in our body in the intensive care unit. Why we want to keep the plants like in intensive care unit? That is called plant tissue culture technology. Artificially, we are growing into the plants in the medium. We are providing the light. Also, the room should be aseptic condition, 100% aseptic condition, free of plant pathogens, free of microbes. Also, the room should be air conditioner, maintaining the relative humidity 80%, and the room temperature keeping for 26 plus or minus 1 degree Celsius. This aseptic condition, this artificial environment, this controlled atmospheric condition is growing the plantlets. Why we want to grow the plants? Why we want to multiplying? I told already, in conventionally the planting material, the farmers are getting from different source with the diseased material, also low yielding. If you go for this plant tissue culture technology, disease free planting material, and also year round we can get the seedlings. That is the foremost important for propagation of plants through tissue culture. Okay, this is the thing we are doing our company. So before that, we never want to forget the inventions. Okay, in the year of 1898, the man, that is the Haberland, he is the father of plant tissue culture. Father of plant tissue culture. Why? The science began. He is the man first. He isolated the cells cultured in the medium artificially. Yes, we can grow a cell into the medium. So that's why he called us a father of plant tissue culture. Similarly, the father of micro propagation. What is meant by micro propagation? If you keep a single shoots, if you keep in a single shoots, that shoots give more number of shoots. 1:5 ratio, 1:8 ratio, 1:9 ratio, like that. He is developed in the year of 1946. One single plantlets enough to develop a 1 lakh plantlets. That is for micro propagation under aseptic condition. Okay, so he is the man also the behind internationally, both the father of plant tissue culture and father of micro propagation for multiplication of plants in the artificial medium, in artificial environment. Okay, so totipotency is the one of the main concept. If you take any part of the plant, we can produce a whole plant. Any part of the plants like stem, root, bit, and also pollen, and also even stem cells, any part of the plants, if you keep in the medium, we can develop a whole plant. That is the concept totipotency. That's why the plant species, we are mass multiplying. Okay, next coming slides, I am explaining how we are doing for tissue culture. So we are the company in the year of 2011, we are incorporated, we are getting the seed license. Also we are the certified for Commission of Horticulture and Plantation Crops. Also we are doing our material for virus indexing, that's for virus testing and genetic fertility. Also we have import export license from Government of India. Okay, this is our nature of business. Mainly we are concentrating for propagation of plantlets. Okay, so what are the plantlets means? First, the foremost important, the important commercial fruit crops is banana. Yes, in globally in India is the production and productivity, we are the #1 producer of the banana. Also we have diversified 1,000 varieties, we have 1,000 cultivars we have. From this 1,000 cultivars, only for the 14 important commercial important banana cultivars are available. If you see in South India, we can see so many bananas. If you go to the North India and in Delhi and Rajasthan and everywhere, the Cavendish group only the green bananas are there. If you come South India, we are getting so many varieties. So we are having granine, red banana, Udayam, hill banana, and Karpuravalli, Rastali. Even we are taking chips, banana chips. That is called Nendran. So we are taking the banana shoots. In farmers, they will take the suckers and cultivate. So what we are doing, we take the high yielding elite clones, we test the virus, presence of the virus through polymerase chain reaction. Then we go for taking the shoots, isolate the shoots and develop the plantlets. That is a mass multiplication. If you keep one shoot, will give 15 shoots within the 2 months of period. So then we are virus indexing. Then these plants will be go for hardening process. Okay, then this is the final product what we are selling to the farmers. Okay, this is a holistic process for the banana. So we are concentrating on banana. Apart from banana, we are doing for bamboo, ornamental crops, floriculture crops, even fig, even orchids. Orchids also we are doing both the dendrobium and phalaenopsis species. Okay. So first we are going to see about our laboratory. So this is the cultures. This is the inoculation chamber. We have the 19 technicians. They are working from morning 9 to evening 6 o'clock. Per day they have, we are allotting for per technician 5,000 plantlets for subculturing, 5,000 plants for subculturing. So we are maintaining the aseptic condition. Our production capacity is 2 million plantlets per year. Okay, so this is the cooked medium we are subculturing. So this is the room we have for the aseptic condition. We are maintaining also scientifically balanced diet, chemicals and growth hormones, controlled growth hormones with the less number of cycles. We are producing large number of plantlets. That's the 2 million plantlets per year in our companies we are doing. So how we are doing? First we are taking the suckers from the elite clone. So high yielding and disease free elite clone, we take the suckers. From the suckers we remove all the microbes, we keep into the artificial medium. Okay, we keep in the artificial medium. This medium, one shoot should be produced 1:10 ratio, 1:15 ratio, the period of 9 months. We are getting a plantlets. So here, high quality, disease free, this is tested for the virus. We are multiplying in the laboratory condition. Once the plantlet is ready, whole plantlets, we go for hardening. The process is called hardening, means from the laboratory condition to in the outside environment. We are accumulating the plantlets into the 26 degrees Celsius. The plantlets is coming out normal temperature, that is primary and secondary hardening. So we are well developed root system. The plantlets are transferred to the polybag condition. We are selling to the farmers. We have more than 13 banana varieties. We have also we are tested. Okay. So apart from banana, we are doing other ornamental crops. Why we are giving importance to the ornamental crops? Now we are facing for oxygen deficit. Everywhere we are, all the peoples are, all the policy makers are talking about environment safety, environment safety. This highly polluted due to the automobile and industrial evolution, we are getting highly polluted. So these are the plants we are multiplying to giving to the vertical gardening for indoor air quality plantlets. I think NASA has studied more than 13 plantlets. It is for cultivating in the Mars for production of oxygen. But on our earth we need these ornamental plants. We are doing and syngonium. We have syngonium 5 types, also syngonium, money plant, and alzondus, spathiphyllum, aglonema. So these are the plantlets for aesthetic value as well as increasing the carbon dioxide amount of the air, as well as so to absorb the polluted air, the pollutants, the toxic pollutants, they will capture into the cells. Is the highly, now the peoples are also, the vertical farming peoples are asking. Apart from, you know, the bamboo also we are doing for the bamboo mission of India. They are also talking about for oxygen park. Nowadays everybody's, they are the NGOs and environment interested scientific communities, they are asking for the bamboo also. The demand now also high biomass and fast growing these plantlets. So we are also doing for the bamboo. So once the plantlets are ready, we are hardening. So we'll send the final products like that to the farmers. We are sending our plantlets to all over the India. Also, we are importing the stage. This is the stage is called ex-agar stage. Ex-agar stage means how the baby is born. Immediately the baby born. That is from our bottle to the laboratory bottle to outside environment. This plantlets we are exporting to the other country also. Okay, we have importing exporting codes. So this is the holistic process. It will take, the process is 10 months. If everybody is thinking about the propagation or plant tissue culture industry is very easiest process. No, it is a very toughest process. It's involving for electricity, manpower, also the time period. If you want to produce a single banana plantlets, it will take 10 months to cross into the different stage. Shoot isolation, multiplication, and also shoot separation and rooting and primary hardening, then secondary hardening, then well matured plants to the farming community. Similarly, this is for 10 months, bamboo is 6 months, ornamental 3 months. Based on the plant species, the propagation time is differing. Also the combination, we have more than 28 crops protocols we have. Okay, so we are stabilized and optimized protocol. We have in scientifically we are multiplying different kind of plant species. Now we are initiated in medicinal plants also cell culture also we are initiated for conservation of medicinal plants, also for extraction of novel components. That is our recent developments. So this is the stage for sending to the plantlets to other states. We are sending plantlets to the Assam also. So through truck also through air also. So if you keep one box, we can more than 1,000 plantlets we can keep in your one box, 1 square feet box. If you take 10 box, in 10,000 plants easily we can take to Tamilnadu to Assam in North India. So within one night journey, they will harden it, they will cultivate. This is the stage for net pot and agar stages. Okay, so this is our final product. In our farmers, we are distributing the farmers from all over the India as well as in Tamil Nadu in hotspot of Theni and Satyamangalam and Tiruchi belt. So and also not only we are doing for Cavendish, apart from this green bananas, we are doing our Yalaki, that is Neipu one, and also Nendran. One of the important banana is for everybody's know Virupakshi, that is high self life, that is a medicinal banana. That also we are doing. We have right now the plantlets. Okay, so this is our nursery facilities for primary hardening. This is the secondary hardening. Also we are using low EC, high enriched compost. Okay, the composts are well decomposed, low EC, and also enriched FOM, enriched FOM with a microbial based enriched FOM. Also we are well developed root system. That is the foremost important. Okay, the well developed root system, well developed plants to the farmers for reduce the mortality rate of the farming communities. Okay, apart from our commercial aspects, we are showcasing our planlets to the conducting a scientific stall, also conducting a scientific trainings and consultancy. Also we are providing the technical support to the farming community. Also apart from this, we are also having artificial intelligence technology app specifically for banana. This also farm advisory service. Immediately the farmers can get, this is the also Englishly available. Immediately the farmers can able to monitor the crop, that our crop is healthy, 95% healthily your plantlets growing. So based on the growth monitoring, we can apply the nutrition. Also we can detect any part of the banana plants, you can scan it immediately. They will tell your plant is 100% healthy, your plant is affected by virus, your plant is affected by bunched up, your plant is affected by fusarium wilt. So like that we are having developed the app. Okay, this app is available freely in Google Play Store, specifically for detection of pest and disease in banana. That is for Tumani, Tumani. If you type in the Google app, you will get the app is freely. Okay, this is the farm advisory service. On farm testing, immediately the farmers can able to monitor the crop. This is that we are applying the recent advanced artificial intelligence technology also. Okay, so this is the overall about how we are propagating a banana ornamental plants. Okay, so this is the holistic view. So now what we are planning now, we will showcase our incubation chambers. Okay, what are the crops we are keeping, how we are keeping, like that. Just we are showcasing 2 to 3 minutes, then we will start for a discussion section. Yes, madam. Okay, I will go now to the laboratory. Yeah, wait a minute. So I will connect through video mode in Sankar Biotech. 1 minute. 1 minute, sir. Dear viewers, now everybody is seeing the screen. Lakshmi madam,
we are yet to see
Lakshmi
Dr.
sir, you are viewing the screen.
No. We are see only your address there. The slide is showing only visit Sankar Biotech. That's the only slide that is being seen here.
Okay, yeah. So
Please
this is
share.
our facilities. So look there, the room is 100% aseptic. We are conducting the air
Dr.
conditioner. So
Sivalingam,
we need
excuse me.
80% relative
Dr.
humidity. To
Sivalingam,
grow your
excuse me.
plants, first and
Dr.
foremost important,
Sivalingam.
80% relative humidity. Room temperature should be 26 plus or minus 1. Okay, this is the facilities. So our
Dr.
laboratory is
Sivalingam,
100%
do you
aseptic.
remain?
So now I am showing for the syngonium. Okay, this is the plant list syngonium. This is the one of the indoor air quality plantlets. From the syngonium, we have 4 to 5 varieties we are multiplying. So like that, we are providing the artificial light and the cultures. Okay, if you see,
Dr.
this is
Sivalingam,
the variegated type. Okay, we have the
Sivalingam,
marble
hear
and
me.
variegated type. Okay, so like that, you can see the banana plantlets also. So see the bananas and ornamental plants. Okay, so you can visibly see these cultures how we are maintaining. See the medium. Okay, so this is the initial stage. For first stage for shoot
Dr.
induction
Sivalingam,
and shoot multiplication. The final stage for rooting. Okay, how the plants we are multiplying, that is the propagation. One shoots will give 1,000 plantlets. Okay, so like that only we are keeping the plantlets. So just see, we are having the bamboo also. Also, we are doing for the orchids. So this. So Planoopsis orchids. This is the one of the
Dr.
important commercial orchids. This also we having Planoopsis and Dendrobium. Also we have the Spathiphyllum. This is the Spathiphyllum. Is also indoor air quality plantlets. So also we are having the bamboo. Okay, this is the bamboo shoots. So how the bamboo shoots are multiplying. So we are keeping the bamboo. So even see the marble type of the money plant. Okay, so also we have the golden money plant, marble type of money plants we have. So like that only we are culturing the plants. You can see the visibly for the banana also. So this is the final stage for the banana. You can see the
participant, are you able
roots.
to
This is
see
the
anything?
complete plantlets we are keeping. So we have the 13 banana varieties. The foremost important is labeling. Okay, the labeling is the foremost important. So see, this is the red banana. Okay, so the red banana, you can see
Not
the
seeing anything, sir.
multiplication,
We are not seeing
also the
anything. We are
rootlets.
hearing only
The
speech.
foremost important is
No, we are not
to
able to see anything. He says he's showing the lab. Dr. Sivalingam, are you hearing me anything? Are you hearing me?
hello sir.
Dr. Sivalingam, I have been telling for the past 2 minutes, we are not able to see your lab. You are explaining. I've been shouting, telling you that we are not able to see your lab. You are not,
Sir,
now only the lab has come. Now you can start. Now
Sir,
only
now
we
you
are
okay?
able to see.
Hello, sir. Now
Now
seeing
it is
this.
possible. Now we are able to see. Now you start.
Yeah, yeah. Okay, sir. Sorry for I didn't share that. Okay.
Oh.
So I will start from the last end. So this is the our laboratory facilities. So this laboratory facilities, we are keeping the air conditioner room. The room should be free of microbial population. That is called aseptic condition. Why we are keeping the air conditioner? So the air conditioner is providing for the temperature maintenance as well as the room is relative humidity 80%. If you want to go for propagation of plantlets to tissue culture, you need to keep 26 plus or minus 1 degree Celsius for the room. Also relative humidity 80%. Okay, the first, this is for the banana. Okay, so I am explaining slowly. So in my hand, I am keeping, this is for the banana plantlets. You can see the root system and shoot system. We have the 13 banana varieties. Okay, 13 banana varieties we are having. Apart from the banana, so I am showcasing for indoor air quality plantlets like syngonium, so spathiphyllum, Aglaonema, okay. So variegated syngonium and money plants. So like that only. So now our director is taking care of the plants, so showcasing. This is the bronze syngonium, okay. This is the bronze syngonium. This is for marble type syngonium, okay. Also he is showcasing the red banana. So this is the red banana culture, okay. So 1:15 ratio. So why the variations of the tissue culture? Also the price also differing from variety to variety, means the rate of multiplication is varying, okay. So we have the 28 protocols from different crops, but the cost is differing. This is Senado. Okay, you know the Senado is the biggest requirements of now the indoor air quality planlets. Also, I will showcase the bamboo. Okay, so this is the bamboo culture. See my hand. So you can see the bamboo. Okay, so bamboo multiplications. So beginning stage. Also see these cultures. Okay, this is the laboratory conditions. So also we are keeping for the orchids also we are keeping. 1 minute I will showcase the orchids. Yeah. Yeah. This is the orchids. Okay. In generally dendrobium species getting fastest multiplication, but the Phalaenopsis is the toughest orchids. Okay. The Phalaenopsis species is toughest protocol. So that also we are standardized. Okay. This orchids are highly valuable for floriculture industry. So we are tied up with the Netherlands based company. We are mass multiplying and supplying the plantlets to the Netherlands. Okay, this is for variegated syngonium. Okay, so now you can see in my hands, this is the variegated syngonium. This is our facilities. So keeping for 2 million plantlets per year our production. Okay, so this is the like. So now our technician also working. I will showcase our technician how we are working. So this is our technicians. They are subculturing the plantlets. Yeah, this is the room specifically for keeping separately for the inoculation chambers. Okay, the inoculation chamber also we need to maintaining the aseptic condition. So in the culture room we have to maintaining the aseptic condition is 100%. Yeah, 100% we need to maintaining the aseptic condition. In the inoculation chamber also we have to maintaining the 90% or 95% free of microbial population. So this is also completely covered with no air, outside air also. So this is the chamber we have. So 8 chambers per day. Our having more than 15 technicians are working in the chamber from morning 9 to 5:30, even sometimes 6 o'clock. Based on the work they are working. So see the planlets. So now we are showcasing our media incubation. Okay. So like that only the cooked medium. Okay. The cooked medium we are keeping and incubated. So once cooked the medium, we cannot directly use it. Okay. The medium is will keep in 2 to 3 days. Okay. We need to check the medium is free of pathogens, microbial pathogens, anything is there or what. The quality control is the foremost important. Once you QC'd the medium, then we will give our technician for inoculation. Okay, 1 minute, sir. Now I am going to connect to the laptop. So we'll start our discussion section. Okay, sir.
Right. Okay, Dr. Sivalingam.
Yes, sir. We are now joined at.
Yes, Dr. Sivalingam. Shall we go for questions now, or you want to say anything?
Yeah, now we'll for open for discussion for the
Right.
audience
Thank you.
having doubts or
O-
anything.
Okay. Thank you for an impressive and a very interesting presentation.
Thank you.
I now request the participants to ask their questions. We'll also be posting the speaker's contact details in the chat window, so that if you don't have time to take up all the questions, you can contact the speaker also separately after the meeting. Now over to the participants for questions. Yes, the first question will be from Mr. Ajit Chowgule. Yes, Mr.
Thank you,
Ajit
sir.
Chowgule.
Lakshmi sir, thank you. Hello, Shankar sir. Basically I am from Maharashtra. We are growing the sugar cane. Are you dealing with only banana or tissue culture? You deal with, I mean, sugar cane also?
Yeah, so no, based on the farmers' order, based on our stockholders' order, only we are mass multiplying. If you want sugar cane, we can multiply it. Give you. So we are doing bamboo also, no problem for sugar cane also.
As per our requirement, you are doing. And the secondly, sir, another question is this thing. I mean, right? Apart from sugar cane, we are this thing, using the seeds for the soybean and some other this thing, aerophone. So, is it practically possible, and this is it advisable to go for the tissue culture? Because sometimes last couple of years, we have faced a lot of problems with the seeds. We are not getting the proper genuine seeds. This is not the problem with us only; it is everywhere. So if I get a good this thing, I mean tissue culture or something from your mother plant, so whether that will be advisable and economical, sir, for other plants other than banana and sugarcane?
Yeah, that most of the our agriculture crops, like you are setting the soybean pulses. So the seed availability of plantlets, no need for tissue culture, sir. For example, groundnut also everybody is cultivating, soybean is cultivating, but larger quantity. So the seed rate is comparatively for horticulture crops and our agriculture crops. The spacing is very important, sir. Okay, so that's why not advisable for our agriculture crops.
Only for banana and sugarcane this is advisable, or this is medicinal plants,
No, no,
or as
no.
you
So
said,
no,
ornamental
no,
plants.
yeah, no. Ornamental and medicinal plant is possible based on the people order. If you are ordering in sugarcane, we will do it and give you back based
Okay, sir.
on the
One more
yeah.
thing, say sugarcane, as you said, you will multiply. But have you done or can you do some any research? Because right now we have to grow for 9 to 12 months. This is the minimum basic period. So,
Okay.
and secondly, you know, everybody after that the sugar percent should be increased. Luckily, and this thing, I mean, in India we have a sugar breeding institute that is in your Coimbatore, and this one is
Yeah,
in
the
Maharashtra.
SBA, SBA, they're having yeah,
Sugar breeding institute. Actually, they are developing, sir. Sir,
yeah,
but farmers like us, they are not listening. I mean, it's not that they are helping, but they are only proposed to give to the institute or the sugar mills, and from there we have to
yeah,
get it. But unfortunately, the sugar mills, they are stuck up with some other problems of selling the sugars and some other financial problems. So they are not giving much importance of this thing. I mean, they are not giving this thing. I mean, support for us. So
okay.
for getting the from the bidding studio, we are getting facing problems. That's why if you can help us, it's well and good. And secondly, sir, whether you can do some research because we want some improvement. Is it
Yeah.
possible from your side in the
So
variety of the sugarcane?
no, the improved varieties. Okay, in for example, so in SBA they having the improved varieties. That is
ఇప్పుడు
the
నేను
noble can
చేస్తున్నాను
everything. Yeah,
లాగిన్ చేస్తున్నాను
if you getting the planting material, that is our mother source. If you give us, we will multiply it and give back you. No problem. For the research point of view, so we are not having that much time to do the research. Okay, already we did the research. Already we have this optimized protocol. So we are in the commercial aspects. If you want to do any new want to for research and developments, so that is a another way R&D, but this is completely for a commercial based companies. If you want for research and developments, we can do it. If you want to sugar cane, if you give the varieties, which varieties you want, if you give the mother plantlets, we can mass multiply and send back you. That is a no issue.
Because recently there's some developments are going up because of some development from the government of India and all those things. Because
Yeah.
huge potential is for ethanol and some cars also there from automobile industry. That's why now everybody is eager. Now they have learned that they have developed some varieties available outside India also, which are rich in fiber content more than actually normally the sugar percentage should be more. That is what is preferred by the sugar mill. Whereas some other companies are there where this thing biomass, that means fiber content should be more. Like that, some varieties have come up. No, it is. I have heard from this made from outside India. So we are interested in that variety, but unfortunately, the sugar mills they will not support because that is against their motto, because they are will be more interested in the sugar rather than the fiber. So, correct. That will be hard. So that will not be economical from those people. But from our point of view, actually, we want from other this thing, fiber content. So that's why I said whether it is possible from your side. That means we know some other plants are available, but unfortunately, we are being a single farmers. They are not willing to give us. So if it should like you, if you can enter in this, and if you can find out some media, or if you can get some other plants.
Okay.
So
Yeah, I will ask one of my friend is working a principal scientist at Sugarcane Breeding Institute, Coimbatore. I will ask the details. Yeah, which variety having the rich in fiber content. If you provide this gem blossom, we will mass multiply and give back you. No problem.
Definitely, sir, more farmers are interested because after the meeting, taking much of your time, I think other some parties may be interested.
You write a detailed email, sir. You write a detailed email, what your need. So based on, we will do it. No problem, sir. Yeah.
So other than banana and sugarcane, what else we can grow this year? Because getting the genuine seeds is a big problem for all the farmers, not me as not only for soybean, then ear of corn or wheat, jawar. So many rice also somehow.
Yeah.
So whether any other this technology, it is a really good one. Appreciating technology, very nice. Whether we can implement this other than banana and sugarcane in other crops.
Yeah, you can go for medicinal values, sir. Now in South India, especially in Tamil Nadu, they are cultivating for coleus. They are extracting for the novel components, secondary metabolites. The farmers are producing the coleus. The tubers are giving to extraction unit. They are getting for essential oils. You can go like that. For example, palmera oil also. You can go for vetiver. Like that, you can go for essential oils and fragrance. Even now they are in Tamil Nadu, one of the company they are doing for growing a jasmine. They are extracting the jasmine, that is a concrete, that is for fragrance. They are importing this the extraction from the Dubai, other markets, Southeast market. So you can go for alternative way of floriculture.
And that is for aromatic plantation. That is also
Yes.
Other vegetables like pudina, palak, these are the very commonly used. We are getting some seedling from outside, but sometimes we do not get proper yield. Is it practically possible and this is economical if you go with the tissue culture of this pudina, palak, which are the leafy vegetables? Because some people are going for the hydroponics because of this problem, sir. So hydroponic, unfortunately, it is a very expensive and this thing critical technology. As of this thing, if you what I compare to those people are years together working in the soil, they will find it rather difficult. What I seen there. So
now this greens you are saying the mint and other things you can grow in the grow bags like in our nowadays the rooftop gardening are emerging. We are good quality of compost we are supplying. If you buy the good quality of compost, you can grow your mint and other things, medicinal plants. It will come up fastly. Pudina and other things.
So how about the leafy vegetable, which is very commonly used for palak, pudina, and then coriander? It is required almost every day in the kitchen. So
so you can grow it, sir. No problem. You can grow it in the grow bag itself. That palak is coming. So we are in South India. We are growing the palak into our container. Okay. So the only thing is the problem for the good quality of soil. The soil should be rich in nutrition. So otherwise you can go for compost. We are now using the compost, the Kaya compost. We have the bricks, 5 kg bricks we are having. That is we are using for hardening purpose. That compost is helpful for to grow a greens also. No need to go for hydroponics.
That is hydroponic. It is a thing out of question. It's a very expensive and rather it is not that much.
Yes. You can go for composting.
Yeah, because we are years together. We are used with the soil technology. So we are finding it rather difficult. But I want to ask you whether we can get this tissue culture, this thing, seedling, or and it will be economical for going for pudina, palak, and this
No. So no, it is not viable, sir. If you want to produce pudina, I am selling a single plant is 10 rupees, how will you buy? That is a question mark. Even sugar cane also, if you want to sell a sugar cane in single seedlings 10 rupees, so nobody will interested to buy this. Yeah, that is the cost involvement.
Just in video I see that.
Yeah.
This video I see that you have, I think you have facility on this thing. I mean, poly house. So only
Yeah.
you will do the first hardening or second hardening can be done at your end, or we have to go for the second hardening, especially for sugarcane.
Yeah, no, on one more thing also, if you want to do for single budded sugarcane technology, they will take now in Tamil Nadu, they are giving for chopped sugarcane. That is single budded. We can harden it, we will provide you. That is also a way, good best way. Otherwise, you can do there itself. We'll provide the composting, good quality of compost to you. You can chop it there itself, you can do it. I will share the single budded sugarcane technology, SSI technology. That is sugarcane initiative technology. You can do, the farmers can able to do in the farm itself.
So anyway, sir, thanks for a nice presentation, and I will get in touch with any. I have taken your number. I will call you later on, sir, because other party may be. Lakshmi, sir, thanks a lot, sir.
Yes, sir.
Thank you. Next question from Ram Babu. Mr. Ram Babu.
Yes. My question is, why you are limiting only to the this banana? There are so many trees like mango, oranges. What is the limitation for you to do these things, like mango or oranges?
Sir, yeah, the mango and oranges, they are doing, everybody is doing for grafted. Okay, the grafted materials are available now, plenty of grafted materials available. Now the trend is changed. Now the forest biotechnology that the forestry department entered, they are looking for tissue culture derived plantlets. For example, teak. Okay, now the teak and also this bamboo. Now only the farmers, also the government also insisted. Why we are doing banana and other ornamental crops? Based on the commercial value only we are doing this crops. We have not only doing one banana, 13 bananas. Based on the order, based on the commercial viability only we are doing. If you want to get any orders, we can do it. For mango and the citrus, that is for grafted materials available, sir.
What about papaya?
Papaya, now the seed is available, sir. That is one of the best varieties. Red lady is available. Papaya we can do tissue culture, but the seed materials available. Hybrid seeds are available in the market. And generally in the nursery, they will raise the seedlings from the seeds. They are selling directly to the farmers.
Okay.
Yeah.
Thank you.
Okay, thank you. Next question. Mr. Siddhappa, you have any question?
Thank you, sir. Thank you, doctor. Will you be able to supply cardamom seedlings?
Yeah, if you want, which variety you want, based on that, we will do it and give you back you, sir.
No, doctor. Can you able to? I mean, can you have a tie up with Spice Board, and so that you will get a good mother source material, and you can supply us?
Okay, we are from which location, sir?
Karnataka, near Coorg.
Coorg, okay. Also, if you want to identify the best variety, Coorg based varieties, if you provide the explant, we will multiply it and give back you, sir. Banana,
Okay,
banana, and we are also doing for black ginger also. Now we are trying. So black ginger and black turmeric also we are initiated very recently. If you provide the explant source, we will multiply and back you, sir. No problem.
Okay, what's the minimum number we have to give, doctor?
5 to 6 plant is enough, sir. Suckers.
Oh, 5 to 6 mother source. Okay, what's the minimum number of seed?
No, minimum you need how much?
Yeah. What's the minimum number we have to give?
No. What is the minimum he want? He has to buy from you. He's asking.
No, minimum.
Yeah.
So how many acre you want going to cultivate, or 10 plants, 20 plants you need?
No, what is the minimum quantity that to propagate and give him? What is the minimum requirement? What is the minimum one has to buy? That is what he's asking, doctor.
So if you want 50,000 plantlets, we can multiply and give back you. No problem.
So minimum one has to buy 50,000 plants. Am I right?
Yeah.
Only then you'll
Yeah.
viable for you to propagate. Correct?
Yes.
No?
Yeah.
Okay, Mr. Siddhappa, you got the answer?
Yes, sir. But what may be the approximate cost? I mean,
In
Okay.
cardamom, like in banana also, for banana we are selling in green bananas 15 rupees, and other bananas we are selling 22, 23 rupees. Based on the multiplication ratio, we will fix out the cost, sir. No problem.
okay. If you supply mother source, how many months it will take to supply, doctor?
Minimum 10 to 12 months we'll give it, sir.
Okay. Thank you, doctor. We will be in touch with you. Thank you, Lakshmipati.
Yeah.
Thank you. Right. Next question. Anybody else is interested to ask question? Mr. Ajit Chowgule wants to ask again. Huh?
Thank you, sir.
Yes.
Shankar sir, I have one.
This conference will now be recorded.
Hello.
Yeah, the farmers are doing through this that cavity trays. Cavity trays are available. Yeah. One minute. I will show the document how the farmers are doing. 1 minute. I will show this.
Okay, sir. Yeah, thank you.
It is a viable, sir. Farmers can do it. So 1 minute. I will share it. That is for single budded sugarcane technology, sir.
Because in our area, the land cost is very high, sir. It's not practical.
Yeah, you can see. Now you can see the PDF. They will chop it the single bud. You can keep it for the trays, pro trays. Okay. So the pro trays, once it germinate like that, you can get the seedlings. That is the one easiest method, sir. Easiest method. You can chop it the single budded 40 days you will get the seedlings. See, 90 wells are available. 90 plants you can keep easily. Okay, this is for a sustainable sugarcane initiative. The government of India is also it is promoted, and now the Tamilnadu farmers are using, sir. You can try this. Okay, sustainable sugarcane initiatives. You can do the chopper also available. If you get single budded and keep in the tray, then it will 40 days you will get the seedlings. Very easiest method, sir. No need for go to the poly bag. You can directly to plant it.
No, to grow the sugarcane, is it practical this thing? I mean, like grow bag rather than on the fields. Instead of fields, I want to grow in a this is grow bag, because we can add anything, no? What we are not normally what happens now, we are spreading the
No. If you go for a grow bag, the growth, the biomass will less biomass. Root mass is biggest root mass now.
So
You
that is
can.
what I see, especially in our area also. What extra? Normally we are putting this. I mean, what you are shown that small seeds we are putting in the tray and we are growing a small seedling and then we are transferring it into the farms. Secondly, what happens now after some 3-4 months, it gets starts some shooting will come up from 3, 4, 10 something like that. And then what happens now in between we are getting weeding and so many these things, fertilizers and the chemicals we are using. And secondly, then again all this requires a huge land, and the land cost is growing every day here. So the cost of the sugarcane, what yield we are getting in a year, and especially compared with the farms investment, it is not this thing. It is not an economical option. So instead of that, whether if we can, because some people, one of our friend, he's from engineering, he has grown sugarcane on a third floor. He is staying in Mumbai on a terrace. He has grown in a bag. He has taken a small guni bag and this thing jute bag, and then he has put some that cow dung and something a small all sand also thing, and he got some 8 to 10 shoots from that one, sir. And it is almost 7 to 8, 10 feet height, and with some 15 to 20. I mean, he has got all this in between this thing. What is it called?
You want to cultivate in the open field or you want to cultivate in rooftop gardening?
Yeah, I want to go with the grow bag, sir. We think practically possible. So we can put the grow bag anywhere, no? It is not necessarily a big field is required. We can, I mean, watering, giving the fertilizer, fumigation, everything. It will be very easy rather than going to the field.
Okay.
And keeping it at a different distance, because we can keep at a different distance as per our requirement. That means the plant should get a proper sunlight and air.
Okay. But the cost effective, it's a biggest cost effective, sir, for to buy the grow bags and compost mixture and everything. So you need to calculate the cost.
No, sir. Right
So,
now, right now we are also putting this biomass, compost, everything we are giving to the fields. Instead of going to the fields, if we go to grow back, it will be easy. No, just like we are having some rose plants or something, this jasmine plant in our this terrace we are keeping. We can feed it, we can water it, we can use some fertilizers. What it will be easy like that. Suppose in an acre we are almost planting 4,500. So for an acre land, cost will be not less than 1 crore, something like in our area particularly. So that is not an economical option. So instead of that, if I can take some land where we can have some good water is there, and we will grow in the bags, and the bag can be placed as a distance which is advisable with the aqua for this thing, just to get every plant should get the proper sunlight and air. This is this is in my mind, sir. I don't know whether it is practical.
I will ask my friend, senior principal scientist. I will ask he's a physiologist. Okay, so I will ask the cost effective whether it is okay or not. No, whether you are growing the sugarcane for industrial purpose or for chewing purpose.
No, sir, actually for industry purpose. That means we are actually making the jaggery from that. So I want to grow and this thing. Second is after cutting it's easier, because after again what happens now, after harvesting of the sugarcane, it is become a big headache now, because we are not getting the labors, and for the small fields the sugarcane harvest is not practical. So this is both ways. This thing farmers are getting a lot of problems. Either they have to do on their own, get the labor, and give them this thing handsome amount and then get it. So that
I will ask, sir. I will ask detailing. I will ask detailing to my friend. I will communicate to you, sir, whether it is possible or not.
Okay, sir.
Thank you. Thank you. Yes. Thank you, Doctor, for answering all those questions. Thank you, participants, for asking so many questions also. We have now completed the round of questions. I would like to thank the speaker, Dr. Sivalingam Elayabalan, and also Mr. Sankar, the director of Sankar Biotech, on behalf of Agriculture Information, for taking their time to speak with us. I now request the organizer of the meeting, Ms. Rajini Jain, to please close the meeting. Thank you.
Thank you, sir.
Thank you, sir. Thank you.
Thank you, Mr. Lakshmipati. I like to thank the speaker, Dr. Sivalingam Elayabalan, and Dr. Sankar, for taking the time to talk with us. I also like to thank all the participants for their time. The meeting will now be closed. Have a good and safe weekend.
Okay. Thank you, madam. Bye.
Thank you.
You're welcome, sir.